Hi everyone, my name is Suditi Kedambadi, and today I will be presenting my project, a novel antibody-drug conjugate to inhibit glioblastoma multiforme. And this project was done under the guidance of my mentor, Dr. Amy Tarangello. So here's my table of contents. I'll be going over the introduction to my project, materials and methods, the results, discussion, future steps, and conclusion. So before we begin, here's a little bit about me. I'm a senior in high school from San Jose, California. I've always been really passionate about science and discovered that my research interests lie in neuroscience and cancer biology. I began working on this project in July 2024 and recently published a preprint of my paper in the Polygence Research Archive. My project was inspired in the memory of my beloved grandfather, who served as a police officer in India for 35 years before he was lost to glioblastoma multiforme in 2017. So here's a brief introduction to my project. First, we need to know what is glioblastoma multiforme. So glioblastoma multiforme, or GBM, is an incurable brain tumor characterized by aggressive progression and frequent relapses. Although multiple treatments are available, the prognosis still remains extremely poor, with only 3 to 5% of people living longer than 5 years after diagnosis. Next, I'll go over the treatments currently available for GBM. Traditionally, surgical resection is the first course of action to remove the maximal amount of cancerous tissue. After surgery, temozolomide, an oral chemotherapy drug, remains the gold standard of treatment against GBM. More specialized treatments include targeted therapies such as monoclonal antibodies or immunotherapy such as CAR T-cell therapy. To continue, here are some challenges that make GBM extremely difficult to treat as a cancer. GBM tumors grow rapidly and are nearly impossible to completely surgically resect due to their fragile location in the brain. Tumor heterogeneity prevents the elimination of areas of the tumor that are unresponsive to targeted therapies. The blood-brain barrier, or BBB, is a semi-permeable vascular boundary lined with tightly packed endothelial cells that greatly limit the chemotherapies that can enter the brain. Finally, cellular dormancy plays an important role in fatal GBM relapses. Chemotherapies are designed to target cancer cells with overactive metabolism or high rates of cell division. However, studies show that some GBM subtypes are able to evade therapy by entering a dormant state and acting as a cancerous stem cell. Fortunately, the constantly evolving field of nanomedicine has shown great promise on this front. Antibody-drug conjugates, or ADCs, consist of a combination of a monoclonal antibody and mAb covalently linked to a cytotoxic payload molecule. The antibody specifically binds to antigens overexpressed on the surface of tumors, releases its payload molecule, and selectively destroys cancer cells while preserving healthy tissue. In my project, I designed a novel ADC to treat GBM and suggest a method of delivery specific to GBM. In the picture on the left, you can see that while ADCs have existed for multiple other cancers, there's very minimal research on how they can be used to safely target and combat GBM. So here is a summary of the computational tools that I use to determine the components of my antibody-drug conjugate. I use DepMap 2.0, GEPIA, and CTRP. So now I will discuss each tool and the methodology that I used in detail. Step one was identifying genes coding for protein receptors with high expression in GBM. The Cancer Dependency Map, or DEP-map, is a portal created by the Broad Institute that contains clinical genetic data from thousands of cancer cell lines from real patients. To visually examine RNA sequencing data of protein coding genes, I generated a scatter plot comparing gene expression between healthy brain tissue and GBM cell lines. After generating a scatter plot, I was able to set conditions for our genes of interest. Genes of interest included those with high expression in GBM cell lines and low expression in control cell lines, otherwise known as an increased Y value relative to X value on this scatter plot. I reviewed multiple genes with Y to X ratios of greater than 3 to 1 log scale or 1,000 to 1 on a linear scale. Interleukin 13 receptor subunit alpha 2 or IL-13R alpha 2 had a Y to X ratio of 3.15 to 1. A literature search confirmed that IL-13RA2 is already under investigation as a biomarker of GBM. I confirmed expression of genes of interest in GBM tissue utilizing the Gene Expression Profiling Interactive Analysis or GEPIA database. GEPIA is a newly developed web server for analyzing RNA sequencing data from thousands of tumor and normal samples from the TCGA and GetX databases. As seen in the bar plot generated by GEPIA, 13RA alpha 2 is highly expressed in cancerous GBM tissue, cancerous pheocytochromata, and paraganglioma tissue, PCPG, and healthy testicular germ cell tissue, TGCT. The next experimental step involved identifying a payload molecule for the ADC using the Cancer Therapeutics Response Portal, CTRP, by the Broad Institute. CTRP is an extensive database that links genetic, lineage, and other cellular features of a cancer cell to small molecule sensitivity to accelerate drug discovery. Of the tens of screening hits identified, BRD1812 was found to have the lowest z-score of negative 2.83 and the strongest inverse correlation of negative 0.487 with IL-13R alpha 2 gene expression. The resulting linear regression line of the scatter plot produced in DepMap confirmed this negative correlation. Based on recent drug sensitivity analyses, CTRP data, and DepMap data, I concluded that the drug BRD1812 would be a promising payload molecule for an anti-GBM ADC. In this slide, I will discuss the results of my literature review for identifying a good monoclonal antibody to target IL-13 receptor alpha subunit 2. In 2012, a study by Balsnikova et al. Developed Clone 47, a humanized mAb specific to IL-13RA2. Clone 47 bound specifically and with high affinity to the native conformation of IL-13 RA2, but not to IL-13 RA1. A linker is the chemical molecule that bridges the monoclonal antibody to the cytotoxic agent in an ADC. Linkers play a major role in ADC target affinity and side effects and ultimately determine its therapeutic efficacy. The ideal linker should remain stable in the circulatory system and selectively release its payload at the tumor site. Multiple studies suggest that the cathepsin enzymes are differentially elevated in GBM cells. This specific linker, a valine citrulline VCIT dipeptide linker, is a standard cleavable linker that has been employed in many successful ADCs approved by the FDA. VCIT linkers are cleaved by cathepsin enzymes upon internalization of the ADCs by target cells, resulting in traceless release of the payload molecule. So to summarize this diagram, the monoclonal antibody that I chose would be Clone 47, which selectively and effectively binds to the IL-13RA2 receptor differentially expressed by glioblastoma cells. The payload molecule is BRD1812, a compound found to negatively correlate with IL-13RA2 gene expression. Finally, the linker is a VCIT dipeptide linker, which remains stable in human blood circulation and can be cleaved by cathepsins overexpressed at the tumor site. In the discussion, I will cover special concerns and future steps. 3 anticipated drawbacks of this treatment plan or our transportation of the ADC across the BBB, heterogeneous expression of IL-13RA2, and off-target binding. The capillary wall of the BBB has a thickness of 50 to 100 nanometers, while the size of an ADC is about 150 kilodaltons, which translates to roughly 225 nanometers. A potential approach to transporting a large ADC across the BBB would be the use of focused ultrasound, FUS, and microbubble therapy to non-invasively and temporarily open up the BBB. Furthermore, although multiple studies suggest that IL-13Ra2 is strongly correlated with poor prognosis, the remaining 20 to 30% of GBM patients with normal expression of IL-13Ra2 may not benefit as significantly from the treatment. Finally, IL-13Ra2 is expressed at high levels in healthy testicular tissue, which was further confirmed by our analysis in GAPIA. Since clone 47 binds to IL-13Ra2 with high affinity, the cytotoxic component BRD1812 may be inadvertently released into the testes. Fortunately, the blood testes barrier, or BTB, is one of the tightest tissue barriers in the mammalian body and should be able to prevent the ADC from nonspecific binding. So here are some future steps that I plan to take. Future steps for this study would involve creating and testing the ADC in a laboratory setting. Cell viability tests such as MTT assays would be used to compare cytotoxic effects between GBM cell lines with different expression levels of IL-13Ra2, including cells that do not express the gene at all. Cell viability assays would also be used for various other immortalized cell lines to determine if off-target cytotoxic effects are present. If in vitro testing suggests that the ADCs solely target and kill GBM cells, further testing can be done in vivo. Under ethical guidelines, a patient-derived xenograft or PDX of a GBM sample can be surgically implanted into an immunocompromised mouse, allowed to grow, and then be treated using the ADC. In this way, we can safely analyze the cytotoxic effects of the ADC in the presence of a blood-brain barrier. Finally, if preclinical studies support the efficacy of this ADC, the efficacy and safety of the ADC could be further tested in phase 1 and 2 clinical trials with healthy and patient volunteers. In conclusion, glioblastoma multiforme is a malignant and aggressive tumor of the central nervous system characterized by an extremely poor prognosis. Current therapies are successful in minimally prolonging a patient's overall life expectancy, but remain ineffective in the long term. ADCs have revolutionized the field of cancer therapy through targeted drug delivery, yet no FDA-approved ADCs currently exist to treat glioblastoma multiforme. In this project, I designed a novel ADC for GBM treatment based on computational analysis. Usage of this ADC could help prevent recurrence after the primary tumor has been excised via surgery. Future research should focus on evaluating the efficacy of this ADC on GBM cell lines, animal models, and human volunteers. I would like to sincerely thank Dr. Amy Tarangello for her continuous guidance, support, and thoughtful feedback throughout the entire research project. She was truly an amazing mentor, and this project could not have been done without her. I would also like to thank Dr. Basola Alabi, my publishing mentor, for her thorough and thoughtful feedback on my symposium slides. And finally, a huge thank you to Polygence for providing me with the opportunity to explore my interests through this research. Here are the image credits in the order used. I also used BioRender to generate and create diagrams of my ADC. Here are the references for my research. And finally, thank you everyone so much for listening to my presentation today. I'm so honored to be able to present my research, and to learn more about my project and read more about it in detail, please check out the entire preprint paper, A Novel Antibody-Drug Conjugate to Inhibit Glioblastoma Multiforme Progression, available at Polygence's Research Archive of Rising Scholars. Thank you so much.